Next, we'll have Preston Andrews with linking soil health to the nutritional quality of fruit. Preston.
Thank you, Steve. We didn't get the sign changed. It tells you just how late we are in this symposium. Okay, this is going to be a very quick synopsis and summary of several research projects that I've been involved with, plus another project that was conducted by the USDA. We got it. Okay. Alex is going to be happy now. Okay, right. And this is a topic we've heard that, soil biology is really, the new frontier, but also part of that linkage is with the growth of the crop and the plants and the roots that are sensing the soil biology and all the other aspects that are going on. So this, in this presentation, I'll be highlighting some of the work of Greg Peck and of Jennifer Reeve, and also Luke Gustafson, who's doing some of the current research. So, but I want to just start with kind of our scientific forebearers here, because, the idea of soil health and plant health is not a new thing at all. And it really, in the scientific community, goes back to Charles Darwin, who studied earthworms in the soil. And if you haven't read his book, the last book he published a year before he died, it's really worth reading, because the observational ability of that scientist is really highlighted in that book. But we also have the book in 1943 by Lady Eve Balfour, and then especially the book by Sir Albert Howard, Soil and Health. And you can read that quote there. It's right in the conclusion of his book. And he really says the nitrogen cycle, is the link between soil and crop and the leaf and the enzymes. And he's talking about proteins here, but he's talking about enzymes and metabolism in the plant. And that is the foundation of healthy consumers. And more recent book on organic agriculture defines living soil, and you read that there: biologically regulated interconnections in the soil ecosystem that play key roles in maintaining desirable soil and physical and chemical conditions. And so that is the heritage that we are now coming back to study, finally. So I want to highlight these two studies. Now, the first one was a 10-year replicated apple study in the Yakima Valley here in Washington State, and it compared organic, integrated, and apple and conventional apple production systems with Golden Delicious and Gala apples. And the second publication from that was Greg Peck's master's thesis. In this study, we actually had replicated plots on a farm, a commercial farm. The second study was strawberry fields in the Monterey Bay area of California. And in that study, we had 13 matched pairs, which were closely in close proximity to each other with identical soil conditions and cultivars. And that was the paper that Jennifer Reeve was part of. Okay, so this is just going to summarize in without showing the data, but just showing you the differences that we find in some of these phytochemical compounds that are present, that were present in this apple and strawberry study. So for apples, of course, we're storing them. So what I'm summarizing here is both at harvest and storage conditions. So I'm not really showing any statistics, but when we look at antioxidants, TAC, measured as TAC, or phenolics, which, because we all make that mistake. Okay, phenolics, which are defense compounds found in the leaves, but also can accumulate in the fruit. We see that as a percent of organic compared to conventional, the organic generally falls always above the conventional in terms of the concentrations of these compounds in the fruit. This would be both at harvest and after storage. In strawberries, we also measured phenolics and antioxidants and vitamin C, and in that case, we're talking about an 8 to 10% higher levels of these compounds in the strawberry fruit under organic management. Now, recently started to do an experiment with tomatoes, and the reason we chose tomatoes was we could grow those annually in a greenhouse, and there's good genetic information on them. In fact, the tomato genome was just published this month in Nature, and there are a lot of genetic mutants that you can use. In this case, we didn't use any genetic mutants, but we used commercially available tomatoes. And so in the first experiment, the preliminary experiment, we used 3 hybrid tomatoes, were indeterminate type of cultivars, in which we used only fertility treatments. The other aspect of this, which will come with Luke's study, was where we included herbivory, which would be insect feeding. And the reason we're looking at this is because there is this discussion or argument about whether when you farm organically, you are managing the soil and therefore increasing the new phytochemical content because of soil management, or is it because organically farmed crops generally have more presence of pests and therefore stimulate these compounds to be produced? So we want to try to separate that in the second experiment. So that's experiment 2 here, where there was 1 hybrid determinant, and we included green peach aphids as in a factorial experiment. So here the data shows, and this is our fertility applications. So the inorganic fertility application shows that most of the nitrogen is in the nitrite form. A good portion is ammonium, and then a very small part of organic forms. The organic, of course, which was a soluble organic form, mostly composed of amino acids, has a large amount of organic nitrogen and some ammonium, but very little nitrate. Okay, now that's what we applied for the 2 fertility treatments. In the aphid treatment, we used these exclusion cages, and into those were introduced green peach aphids in half of the study. So what we show here is the number of aphids on the aphid-introduced crops, the aphid-introduced tomatoes that were grown either with inorganic fertility or with organic fertility. And we did not find quite at the 0.05% level a difference, even though there's a 4-fold difference in the aphid density on the leaves of these 2 treatments. But you can see what the variability was fairly high. So in these 2 tomato experiments, and in the first one there were 3 cultivars, and this really illustrates, I think, the problem that a lot of this research has, is because if you choose a certain cultivar, you might get a certain answer. If you choose more cultivars, you end up with more confusion because they don't all correspond with each other. And that's what happens quite frequently. And so what we see here is the antioxidants being not significantly higher, even in the organic here, and the phenolics being significantly higher and the vitamin C higher, but the lycopene wasn't, but there was a treatment by cultivar interaction. So in some cases, the lycopene might have been a little higher in the very small fruited tomato cultivar, but it was not in the large fruited cultivar. So there's quite, that has to be taken into consideration. In the second experiment with only 1 cultivar, here you can get clean results because you're only comparing, and if you're lucky, you chose the cultivar that is going to show you the differences that you can publish. But if you choose another cultivar, you may not find that. And so that really illustrates the importance of the genetics in these comparisons. I'll move along quickly here. Okay, so we looked at phenolics also, and this is all based on fertility treatment differences. So we found very little effect of the aphid treatment, except for phenolics in the fruit, shown here. The phenolics in the fruit under aphid infestation was higher than when no aphids were present. So they may have stimulated some response. Soluble solids and dry matter. Again, this is something that can be variable, but generally shows a positive effect from organic fertility management. In the strawberry, 1 of the cultivars showed significantly higher soluble solids content. In the first tomato experiment, no difference. In the second, much higher. And so if you chose the right tomato cultivar and you grew it under conditions in which processors would want to use them, you would make a lot more money with a tomato that had higher soluble solids content. Dry matter, again, it was variable and depends really on the cultivar. A recent meta-analysis was published, and this compared a number of studies. It was a meta-analysis, and there were conditions for inclusion in this. And what they found was that defense compounds are generally higher, 15% higher on average, than in the organically grown fruits, vegetables, and products than in the conventional. And non-defense compounds were only 7% higher, and in some cases may not have been different at all. So this this lends credence to the argument that soil management practices can influence the defense compounds, and that those are the compounds that are of benefit to our consumption. This highlights the work of Jennifer Reeve that she did microarray analysis of the strawberry fields, and in all cases we see that for nitrogen fixation and sulfite reduction and these other aspects of the soil biology, they were all elevated in the organic managed system, and that could be an important criteria when selecting for cultivars that could be grown in an organic farmed system, that they would be appropriate for that production. The last part of this is some work that was done at the USDA looking at tomatoes grown under black plastic mulch or under hairy vetch mulch. And in that case, they looked at the gene activity in the leaves of these two treatments, and they found that nitrogen and carbon-related genes were all upregulated in the hairy vetch managed, as well as chaperone proteins that bring proteins across the cell membrane, and defense compounds were all upregulated. Again, something like chitinase would be really important for protection against insects and for protection against fungi. So they came up with a scheme in which they looked at organic matter decomposition and microbial biodiversity. This is kind of like their model of what is happening, going into nitrogen assimilation here and carbon assimilation both in the fruit and in the leaves, and the protective compounds that are accumulating in the leaves as secondary metabolites. So just to acknowledge all the people that have helped and funding agencies over the years. Thank you.